Hello, my name is Gargi Nandi. I am an 8th grader at Carmel Valley Middle School, and I'm going to tell you about a novel technology for editing DNA called CRISPR. DNA stands for deoxyribonucleic acid. These are long strands of specially arranged proteins found inside the nucleus of every living cell. The strands are essentially sequences of the four building blocks of life: adenine, thymine, cytosine, and guanine. Now, why would we want to mess with these strands? DNA is a set of instructions which tells a cell how to function as a part of an organism. For example, the DNA in our body cells has information on how tall we will be, or if we'll get pimples. More seriously, what diseases we might inherit or have a tendency for. That is why scientists are interested in knowing if there is a possibility of modifying a person's DNA to potentially save their life. That's where CRISPR technology comes into picture. Scientists accidentally discovered this technology when they were studying bacteria's immune system when attacked by a virus. The acronym CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeat. Now, to understand exactly what the scientists discovered, let's introduce the main actors in our story. Let's start with the bad guy. This is Dr. Virus. Now the good guy. This is Special Agent Cas9, or CRISPR-associated protein 9. When a virus attacks a bacterial cell, it injects its DNA inside the cell. Now, the cell has a special defense system called the CRISPR mechanism. The cell sends out some detectives. They are proteins called CRISPR-associated proteins 1 and 2. They scan the virus's DNA for its quote-unquote fingerprints. The fingerprint is a sequence of nucleobases called spacers. The detectives make a copy of this fingerprint and send it back to special locations on the bacterial DNA called CRISPR locus. It is basically a place where the DNA files away this fingerprint by sandwiching it between the repeats of short sequences of bases called repeats. If multiple virus types attack the bacteria, these detectives save a fingerprint of each of them, making a many-layered sandwich. How does a bacteria protect itself when it's attacked again? The bacteria first sends out its special agents, its very own 007s. They are proteins called CRISPR-associated protein 9. These agents are armed with the fingerprints of the viruses that have attacked in the past. These fingerprints are called guide RNAs. When a virus attacks again, the agents try to match up the fingerprints of the attacker with the fingerprints on their file. When an agent finds a match, it snips the attacker's DNA to disable it. What scientists have done is modify this mechanism to disrupt the expression of specific genes in an organism. They have come up with a way of assembling a modified CRISPR-associated protein 9 armed with a guide RNA or fingerprint for specific genes. Scientists are able to insert these agents into the nucleus of any cell, not just bacterial cells. These agents inspect the cell's DNA to find a match for the fingerprint of their assigned gene. When they find a match, they cut the gene sequence out of the DNA. The cell then tries to fix the broken DNA. The errors in doing this cause a mutation in the gene. This procedure is called gene silencing. It is also possible to add a recipe for fixing the disrupted gene, and the procedure is called gene editing. The recipe is the template RNA or instructions for how you want the gene to be modified. This way, scientists are able to achieve the desired gene modification. Experts think that the discovery of this mechanism is a giant leap forward in many fields. For example, industrial bacteria, such as those used in food production, can be modified to be immune to viruses. This will reduce waste and make food processing cheaper. In medicine, gene editing can be used to fight hereditary diseases such as liver disorder. Antibiotics can be designed to attack only specific bacteria and spare the beneficial ones. Won't it be fun to take your antibiotics without upsetting your gut flora? In scientific research, precise editing of specific genes in lab organisms can be used to study the association between a given gene and its effect on the organism. You can learn much more about the CRISPR technology by following the scientific references in the material I used to make this video. My first introduction to CRISPR technology was on Radiolab. I highly recommend starting there. This essay on Harvard University website gives a very nice overview of this technology. Finally, there is Wikipedia and a bunch of reviews in scientific journals such as Nature and Science. I used these resources for images and audio. I hope you enjoyed this video.